A very good afternoon to all of you.
Good afternoon, sir.
Good
Good
afternoon,
afternoon,
sir.
sir. Good
Good
afternoon, sir.
afternoon,
Good
sir.
afternoon,
Good
sir.
afternoon, sir.
Good
Good
afternoon,
afternoon,
sir.
sir.
Good afternoon,
Good
sir.
afternoon, sir.
Good afternoon,
Okay.
sir.
Afternoon, sir.
Uh, am I audible to everyone?
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes,
Yes,
sir.
Yes,
sir.
sir.
Yes, sir.
Okay. And can you see my screen?
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes,
Yes,
sir.
sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Okay. So I will be teaching you the first paper of the syllabus, that is cell biology and genetics. Okay. So the
Yes,
cell
sir.
biology,
Yes, sir.
you are reading from,
Excuse me, sir. Excuse me, sir.
yes.
Sir, it's not visible. Sir, it's not visible.
Okay.
Yes,
Uh,
sir. It's not visible.
is it visible now?
Now,
Now it's
Yes, sir.
yes,
yes,
sir.
Yes,
sir.
Yes,
Yes,
sir.
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 Yes, sir.
Yes, sir. Yes, sir.
Okay, okay, okay. Okay, when I will show the slides, you can see that. Just before that, I would like to tell you something. So, starting from maybe class 7 or 6, you are reading this cell biology, right?
Yes, sir.
Maybe
Yes, sir.
Yes, sir.
later stage you are reading it. You have gone through this course during your high school career, then at higher secondary level, and then at graduation level, and still at post graduation level. Okay, is
Yes, sir.
it essential to study all these things?
Basic things.
Why should we study the cell, right? So whenever you are going through a course, there must be some objective, right?
Yes, sir.
Why should we learn
Yes,
that?
sir.
Will it be
To know the
for
basic.
Sir, the
okay?
sir the slide, sir the slide, sir not visible, sir.
Okay, I have not played the slides, so don't worry. Okay, so why we are still reading this cytology or this cell biology and also this genetics? Okay, because you see, whenever anything what you want to try, first you try at cellular level, right?
Yes, sir.
So you cannot go directly for an organism to
Without.
test the effect of one vaccine or one compound or something like that. Okay, first you go at the cellular level because cell is the fundamental unit of all the living beings, right?
Yes, sir.
Okay, so
yes, sir.
today we'll be dealing with some of the introductory things that you have gone through in your last curriculum, maybe in graduation and in plus two also. So in the next class onwards, we'll go to this basic higher things that will be there. So today, and is it visible now?
Yes, sir.
Yes,
Yes, sir.
sir.
Yes, sir.
Yes, sir.
Yes,
Okay,
sir.
Yes, sir.
okay, okay. So if you look at this cell, what you can find that there are different organelles, and these are embedded inside a plasma membrane, and in some cases there is another cell wall or cell membrane over there, and inside that you have different organelles, right? So uh, you know that all organisms are made up of cells, and these cells working together, similar cells working together, having a common origin, we call them like a tissue. Then several tissue, they form this organ, and several organs they make this organ system, and there are different organ system one organism. This is the level of organization in any kind of living world. And from one organism, when there are several organisms of a particular kind living in a particular area, we call that as a population. And several population forms a community, and uh, there are several community in an ecosystem. And no, there are several kinds of ecosystem, and we call them like biomes. And all living, non-living things on the earth, we call them at that's biosphere. So if you want to study the biosphere even, you have to start from the cell onwards. Okay. So before um, going for any subject, first of all, you should have to have some knowledge regarding the history of those things. So, coming to the history, um, the first person who discovered this cell, that is Robert Hooke, in 1665. He used his own handmade microscope to study these plant cells. Okay, and he had studied these cork cells. Okay, these cork cells. And what he saw like small boxes. This is Robert Hooke. Okay, and he named this cell because they look like the cells those are used by the monks to live. Okay, now this is the next person, that is Anton van Leeuwenhoek, and is a Dutch scientist. And in 1673, he could view these living organisms. See, for the first time, he saw the living organisms. But what about Robert Hooke? He saw only the dead cells. Okay. Now, Leeuwenhoek also used his own handmade microscope to see these organisms in pond water, and the scraping from his teeth. And next, there came a very uh, beautiful period in the history of the cell biology, that came the cell theory. Okay, there are two persons. One is a botanist, another one is a zoology person. Those are credited with the cell theory. Okay, the German botanist Sleden has shown that all the plants are made up of cells, and uh, okay, and then the German zoologist Swan, he also told that the animals are made up of cells. Okay, so both are credited with the theory of cell theory. And then another person, Virchow, okay, he's a doctor, and he could see the dividing cells under the microscope. Okay, and he told that all the cells they came from the pre-existing cells by cell division. Okay, so new cells came from the pre-existing cells. Okay, so these are some of the basic things of the cell theory, that all living cells are made up of cells. Cells are the basic unit of structure and function in an organism, and these new cells they came from these pre-existing cells. Okay, now in cells you will find there are several organelles which have their own genetic material. Can anybody tell what are the organelles that has their own genetic material? Anybody?
Sir, mitochondria and plastids.
Okay, mitochondria and plastid or chloroplast?
Chloroplast.
Okay, thank you. Now, this earlier it was thought that these organelles, these mitochondria and chloroplast, they are like some kind of prokaryotic organisms. They are free living organisms, and they came inside these cells. So this endosymbiotic theory was there, and after that, these organelles, these organisms were uh, remain inside this cell, that is called your chloroplast and mitochondria. Okay, so this endosymbiotic theory tells about these semi-autonomous organelles. Okay, now this is how these mitochondria, okay, they came to the cell. Okay, this plastid and these are your mitochondria. Okay, now coming to cell size. See, cell has a very small size. Do you agree?
Yes, sir.
Yes, sir.
Why?
Yes, sir.
Yes, sir.
Yes, sir.
Why? Why cell has a small size? Of course, there are certain cells which are visible to your naked eye, just like the very big egg cells or some plastids or the plant cells. Okay, but normally the cells are very small in size. Why? Can anybody tell? No? Okay. Anything that is coming into the cell or coming out of the cell, how they are by which means they are coming in and out? Through the
Cell membrane.
Cell membrane.
very good, very good. And the rate of this transport will depend on what? On the total surface area, right?
Yes, sir.
Okay.
Yes, sir.
So if the surface area of the cell to its volume, okay, if you take the ratio of the surface area to the volume of a cell, and if you will find that, let us take one cell. Okay, I can show you, just 1 minute. Okay, so if let us think that the cell is a cube. Okay, cell is a cube, and each side of the cube is say 1 unit. Okay, so what is the volume of this cube now? Who can tell? There is a cube, and the side of the cube is 1 unit in length, maybe 1 micrometer or 1 cm, something like that. So what is the volume of that cell? Anybody?
1 into 1 into 1.
That is 1 cube.
1 cube.
Okay, 1 cube unit. Okay, now what is the surface area of that cell? So how many
1 square.
how many surfaces are there in a cube? There are 6 surfaces, right?
Yes, sir.
So the total surface area is 6 into 1 square, right? That is 6 unit, 6 square unit. Okay, so if you take the volume is to your surface area, what will happen? Uh, what is the ratio? That is 6 is to 1. Okay, now let us change the side of this cube as 2 unit. So what will be the volume?
8.
So
8
it
cube.
is now it is 6 is to 1, right?
Yes, sir.
Initially it is 6 is to 1. Now you change the side of the cell to like the cube to 2 unit. So the volume becomes
8
8.
cube unit.
Okay, 8 unit, 8 cube unit. Now what is the surface area?
24.
24.
24. Now what is the ratio? 24 is to how much?
24 is
Sir,
to 8.
8 is to 24.
8.
So 24 is to 8. That means how much it becomes?
3 is to 1. 3 is to 1.
3 is to 1. Very good.
One.
3 is to 1. Now let us make it as 3 unit. So what would be the volume?
27.
27.
27.
27. And what about this surface area?
54.
54, sir.
So what is the ratio?
2 is to 1. 3. 2 is
1 to 1.
2
27
is to 1.
4, 1. 2
Now
is
make
to
it
1.
now make it 4.
64. 64.
So what is the
So
volume
1
of
is
the
to
cell?
1.
64 cubic units.
64 cubic unit,
Yes,
right?
sir.
Yes, sir.
Now what is the area?
64.
64. Now what is the ratio?
1 is to 1.
1 is to 1.
So as you go on increasing the volume of the cell, what will happen? The surface area is reducing. The relative surface area is reducing, right?
Yes, sir.
Did you
Yes, sir.
get that? Did you
Yes.
get that? When it
Yes,
is
sir.
Yes,
1,
sir.
this the surface area is like 6 times more. Okay, and when the size increases, what happens? The surface area is going and decreasing and decreasing and decreasing. Relative surface area. Did you get
Yes, sir.
that? Okay,
Yes,
so
sir.
Yes, sir.
in order to control the like transportation of the things from the cell to outside or from outside to inside, they need to have a higher surface area. Right, the surface area is larger, the rate of transportation will be better.
Hi.
Okay, so for that reason the size of the cell is small. Okay, did you get that? Why the
Yes, sir.
size of the cell is small? Okay, now as you know, there are three kinds of cells.
Sir, there is some glitch. Can't hear.
Normally, you find it is maybe a circular in structure, and this plant cell which is polygonal, and this bacterial cell having different type of its. Okay, now coming to this number of cells, there are certain organisms which are unicellular organisms, right? And there are certain organisms which are multicellular organisms. Can anybody tell any unicellular organism name of any unicellular organism?
Amoeba.
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 Okay, any other? So an amoeba is a plant cell or animal cell?
Amoeba.
Hmm?
Animal. Animal cell.
Can you say any it's a like a prokaryote or something eukaryote? Can anybody tell?
Prokaryotic cell.
Okay, what? How can you say? How will you able to say one cell as prokaryote or eukaryote?
So
Suppose
prokaryote.
I will show you 3 or 4 cells under microscopes, right? Okay, I'll keep 4 5 cells under microscope. How can you say which one is eukaryote, which one is prokaryote?
Sir,
The
by the
Sir,
prokaryote do
nucleus.
not
eukaryote
have a
has a
distinct nuclear
well-defined
wall.
nucleus, whereas prokaryote does not have a
Members are
well-defined nucleus.
having no nucleus. In
Very
prokaryotic cells.
very good, very good. And how can you say which one is a plant cell and which one is an animal cell?
Presence of
Cell,
cell wall.
Cell wall.
Presence of
presence
cell.
of cell wall.
So whenever by looking at the shape of the cell, you can say whether it is an eukaryote, prokaryote, sorry, animal cell or a plant cell. Okay.
Yes, sir.
So in case of unicellular organism, you will find it is composed of only one cells. Okay. Whereas in multicellular organisms, you will find there are several cells in this organism. And see, the size of different objects or things that is there in a cell to the like organism, you'll see the size of this atom, then your complex molecule like amino acids, proteins, then your viruses, then your organelles like chloroplast or some bacteria, then plant animal cells, then human egg, frog egg. Now see how these cells, the size of the cell is going on increasing. Okay, so typically the range is from 5 to 50 micron in diameter. Okay, these are very basic things. Of course, you know these things. So this is a plant cell. The size is more than that of the animal cell, which is more than that of the bacteria or prokaryotic cell. Okay. Now coming to the cell and its special functions, there are different kinds of cells, and they have different kinds of functions. And these cells, they form this tissue, and you see these are like blood cells, and you know blood cells, right?
Yes, sir.
What are
Yes, sir.
different kinds of blood cells? Anybody can tell?
RBC, WBC,
RBC,
RBC, WBC, and blood platelets.
What is WBC?
white blood cells.
white blood cell,
White
Blood
blood
cells.
cells.
lymphocytes,
And okay, very good. And what is their
leukocytes.
functions? What?
Immunological function.
Helps in phagocytosis.
Okay, very good. And it gives us immunity, right?
Immunity.
Yes.
Yes.
You know there are like neutrophils, basophils, right?
Yes.
Yes,
Yes,
sir.
sir.
Yes.
Okay,
Eosinophils.
eosinophils, right?
Yes.
Yes,
Yes.
sir.
Yes.
Okay, and you know these macrophages, the dendritic cells, right?
Yes, sir.
Yes,
Okay,
sir.
then you know T cell, B cell, you know. Okay, maybe you will be learning all these things in due course of time. And you know there are certain cells which are responsible for this uh, function of like say communication, like these your neural cells, this nervous system. Okay,
Yes, sir.
and there are some muscle cells that helps for this mechanical work, right? So they have different special kind of function. Just like this is a muscle cell, and this is what kind of cell this is? Can anybody tell? What is the cell?
eosinophil,
Is it eosinophil? Is it
Mm,
a neutrophil? Okay, this is a neutrophil because this neutrophil are polynucleated in structure. Okay, just like this. Okay, so from this you can say, and this is a neural cell. Okay, so sometimes these cells, they are forming special kind of functions, and this special kind of function is uh, performed by the cell after the switching on and off of different kinds of gene present in the cell. Because when the organism started, when the creation of the organism started from a single cell or from a zygote, you know, it was a single cell, and after subsequent cell divisions, some cells, they become specialized as neural cells, some become like blood cells, some muscle cells, something like that, right? And this process is called differentiation. Okay, can anybody tell what is the disease that we suffer from when there is poor differentiation of the cell? In which kind of disease there is a poor differentiation of cells?
differentiation of cell.
Yes.
sickle cell anemia.
No, in sickle cell anemia, it looks like a sickle, right? And in case of when somebody is suffering from cancer, you'll find that the cells are what, they are poorly differentiated. They don't differentiate at all. Okay.
Yes.
Now, coming to these muscle cells like this, and your red blood cells and your cheek cells. Of course, when you will be doing the practicals, because things are happening normal, we are going towards normalcy after this pandemic. So probably you will be joining us in the offline classes, and then we'll be doing this practical to see the cheek cells. Okay, and there are certain cells in case of plants also. These are the case of specialized cells in animal system, and these are some cell in the plant system, your guard cells. And yes, these are your xylem cells, these are your metaxylem, these are your protoxylems, and then these specialized pollen cells. Okay, where pollen grains will be there, and these are your organization of the cells, how these cells are created. You see, first from these your elements, compounds are formed, and then you see these amino acid and the complex molecules are formed, and after that you find this DNA, amino acid, and after that this uh, organelles are formed, and from organelles you could find these cells and then these cell systems. Okay, so from organ systems to different organ working together, and then to your organisms. Okay, yes. Now there are, as I told you, there are simple cells, unicellular cells, and complex cells or multicellular organisms are there. So in case of these prokaryotic cells, there is no clear-cut nucleus. There is a maybe single circular DNA or nucleolide-like things, and this is very simple. And the chromosome is single circular. Okay, this is a prokaryote, and this is your nucleate, and this is the cytoplasm, and these are the capsule. This is the cell wall, and sometimes these are the ribosomes. What kind of ribosome are there in prokaryotic cells? What are the kind of ribosome those are present in a prokaryotic cell? Anybody?
So 70S.
70S.
70S and
Okay.
Yes, 7.
Very good, very good. There are 70S ribosomes in this prokaryotes, whereas in eukaryotes you have 80S kind of ribosomes, right? Can anybody tell what is this S stands for? What is this S?
The sedimentation
Step by
coefficient.
unit.
Sedimentation coefficient.
unit sedimentation.
Sedimentation coefficient or Seidberg's coefficient, right? And you see, this is eukaryote. You will find the eukaryote having a clear-cut nucleus. Okay, and it includes this fungi, protists, plants, and animals, and these are more complex. When coming to eukaryotic cell, you have three basic structures, like you have a nucleus, then your cell membrane, and cytoplasm with these organelles. Okay, now as we are talking regarding these both type of cells, this plant cell and animal cells, you see, in case of plant cell, you have a very large vacuole, right? Okay, what is the membrane of the vacuole known as? Anybody can? Very,
Monoblast.
very good, very good. And these organelles are microscopic in nature, and they are found in cytoplasm, and sometimes they are membrane bounded, and sometimes there are certain those are not bounded by any kind of membrane, like your uh, certain organelles. And this is animal cell, and here you don't see any kind of large vacuole that is there in case of these plant cells. Okay, and this is a plant cell, and this is the central large vacuole. Can you see the large vacuole?
Yes, sir.
Okay, now, and this is this plasma membrane, and you see in the plasma membrane there are certain things like proteins, and there are certain sugar molecules attached to that. These are your glycoproteins, and these are your carbohydrates. Okay, and this is lipid bilayer. So while describing the plasma membrane, we'll study all these things. Okay, and this is double layered structure containing certain proteins. Okay, and they surround all the cells, and this is also responsible for the transportation of things to the cell and out of the cell. Okay, so this is a living layer. See, so this is phospholipid that is there in the plasma membrane. It has a hydrophilic head. Okay, this is hydrophilic head and hydrophobic tail, right? So these tails are made up of fatty acids. So these are hydrophobic in nature. And I see there are certain proteins associated with this. And these proteins may be intrinsic protein or extrinsic proteins are there. We'll discuss that in later stage. And through this membrane, you can find that these oxygen, carbon dioxide, and water they can move through. But for the in transportation of certain larger molecule, you need certain kind of carrier proteins. We'll be discussing that. And this is how these intrinsic proteins are there, and these are your hydrophilic heads, and these are your hydrophobic tails. Okay. Now this is how the proteins they help in transportation, and there are certain proteins we call them like peripheral proteins. They're attached to the surface. Okay. You know, there are like certain receptors over the cell membrane that can recognize different compounds, maybe ligands. Okay. So there are integral proteins. Those are inside the membrane. Okay. Throughout the membrane, those are uh, integral proteins. And so this is one glycoprotein, where you will find a protein is uh, attached to the uh, carbohydrate. Okay, these are carbohydrate molecule. Okay, so when a protein is associated with this carbohydrate, we call them like maybe some glycogen or something. So we call them like glycoproteins. Okay, and glycoproteins have carbohydrate tails. Okay, and these are responsible for cell recognition. And in case of cell membrane in plants, this is the cell membrane that is against the cell wall, you know, and this gives a shape and maintains the cell shape. And the cell wall is non-living layer, and it is found in fungi and bacteria, and this is made up of cellulose. And whereas in case of bacteria, you will find this peptidoglycan is there, and chitin in case of fungi. So this is the cell wall that is present in plant system. Okay. Now coming to this cell wall, what is the role? It supports the cell, right, and protects the cell. And this found up outside of the cell membrane. So this is the cytoplasm, and a jelly-like substance that is there inside the cell membrane, and it provides the medium for chemical reactions. Okay, what are the different chemical reaction that is happening in cytoplasm? Can anybody tell which one is the most important thing that is happening in cytoplasm? Anybody?
So glycolysis.
Glycolysis. What is the most important function? I'm asking. Can anybody tell where protein synthesis takes place? Hmm?
Ribosome.
Ribosome.
Very good. And where ribosome is present?
Cytoplasm.
Sir, endoplasmic reticulum.
Okay. So
Attached to the endoplasmic reticulum.
very good. There are two kinds of ribosome on the basis of their like location. One is attached to your endoplasmic reticulum. We call them like rough endoplasmic reticulum, right?
Yes, sir.
When the ribosome get attached at the endoplasmic reticulum, we call them rough endoplasmic reticulum. And there are certain free ribosomes, those are present inside the cytoplasm also. Okay, so they are the site of protein synthesis, right? I hope protein synthesis is the most important thing that is happening inside the cell, other than this you say there is your plastid where you can find this dark reaction, light reaction is happening, right, in chloroplast, right?
Yes, sir.
In stroma and grana, and similarly in mitochondria you will find different kind of respirations are taking place, or in peroxism something is also taking place, right? So these organelles, they carry out specific jobs. So these things are done in cytoplasm, okay? And this cytoplasm is omnipresent, like in all cells you can find this cytoplasm. And now the most important organelle is your nucleus, right? Why? Why nucleus is the most
Contains the
important organism or organelle?
contains the DNA.
Okay, so what? It contains DNA. So why it is important?
Cell division.
It's a genet.
Yes.
So
So
it
it is
contains
genetic
genetic
unit.
Sir, it
material.
contains the genetic material.
See, whatever we are doing and what is our phenotype, what is our character, everything is encoded in our genes or in our DNA, right? So what the cell will perform, what will, how it will function, everything depends on this information that is present inside the gene, okay? And that is present inside the nucleus, right? So how the cell will function, what will be its morphotype, and to what it will be transformed, okay? Or what will be its differentiation, all these things depends on these genes, okay? These coding regions in your nuclear DNA. So they decide the fate of the cell, okay? So for that reason your nucleus is your what, controlling center of the cell, right? And when the size of the cell increases and there are certain other things happening uh, in cell or around the cell, you will find the cell try to divide or may remain in an like in a resting stage, right? So all these decisions, those are taken by the nucleus, okay? So we call them like controlling center of a cell. And this nucleus was seen by Robert Brown in 1831, and this is bounded by a nuclear envelope, okay? And this nuclear envelope has certain pores, okay? There are certain pores like this. These are called nuclear pores. Of course these are nuclear pores here, you can find out, okay? And to this your endoplasmic reticulum is attached, okay? Now it is the largest organelle in the cell, okay? And this cell has a fixed number of chromosomes, okay? And that carry genes. How many chromosomes human beings have?
26.
20.
23 pairs.
23 pair.
23
23
pair.
pairs.
23 pair or 46 number of chromosomes we have.
46.
Okay. So out of that 46 chromosome, your 40 chromosomes and the rest two are called your sex chromosomes. Okay. So
Sex chromosomes.
sex chromosomes. So your character, your personality, your morphological feature, everything depends on these genes. Those are present inside these chromosomes. Okay, and depending on different kinds of genes, those are switch on or switched off. You are suffering from different kind of genetic disorders also, right? So this is your nuclear envelope, and these are your nuclear pores. Yes, these are your nuclear pores. Okay, so this is a double membrane structure, and we call them like nuclear membrane, and there are nuclear pores. Can anybody tell what are the thing those are coming out of the nucleus and what are the thing those are coming inside the nucleus? Can anybody tell? Through the nuclear pores, something is coming outside the nucleus and something is going inside the nucleus. Can anybody tell what are the things that are going out and going into the nucleus?
Sir, the DNA from RNA, the replication of DNA from RNA, the RNA which is synthesis from the
Okay,
DNA,
this RNA is produced.
The RNA molecule.
the
Yes,
RNA is
that is
came from
coming out of.
the
Okay, so it is coming out of the nucleus to the cytoplasm where the protein synthesis takes place. Okay,
yes, sir.
can anybody tell why there is why this DNA or this nuclear material is present inside the nucleus, not outside?
Because it is the membrane bound organelle.
No, nucleus is the membrane-bound organelle. That is fine. Why this genetic material, that is your DNA or this nucleic acid, that is present inside the
Nucleus.
nucleus, not in the cytoplasm? What will
Like
happen?
nucleus is
What
principal
will
organ of.
That is fine. What will happen if this DNA or this nucleic acid that is coming out of the nuclear membrane or out of the nucleus? Can anybody tell?
No, sir.
So where is the lysosome present?
Cytoplasm.
Very good. Lysosome is present in the cytoplasm, and also in the cytoplasm you'll see that there are several types of enzymes like your endonuclease and exonuclease are there. Those are present in the cytoplasm. So what will happen if this nucleic or nucleic acid or this chromatin material or that is your DNA that is comes out of the nucleus? That means
If they
the-
if
these
they
these nuclease, endonuclease or exonuclease, they
they
will what
can they
they
can
will
they can lysis the
they
breakdown
will
of
chew
the
the DNA. They will cleave the DNA.
bleed. Yes, sir.
Okay, they will cleave the DNA. So for that reason they are not coming out of the nucleus, and when they are coming out of the nucleus they are processed, right? You know there is a poly-A tail and there is a G capping, right? During protein synthesis we will be starting at in molecular biology. So that is there, so that can safeguard from these different exonuclease and endonuclease. So when this RNA that is coming out of the nucleus, those are not cleaved by these enzymes. Got it? Okay, so this genetic material that is DNA is found inside the nucleus. Can anybody tell what is a molecule of DNA? What is a DNA molecule? Can anybody tell? Can anybody tell what is a DNA molecule? Anybody?
Nucleic acid.
Okay, it's nucleic acid, fine. But if I'll say one molecule of carbon dioxide, that means CO2, right?
Yes,
One molecule of water, that is H2O.
yes, sir.
Is it? Is it? So what if I'll tell what is the formula of one molecule of DNA?
So nitrogen base, phosphate and sugar.
Nitrogen base, pentose sugar.
So if that is a DNA, then what is like a nucleotide? You know there are three things. One is your nucleoside, right? One is nitrogenous base, and when this nitrogen base is attached with your sugar, that is your pentose sugar, maybe
Pentose sugar.
deoxyribose sugar, we call that as a nucleoside, right?
Nucleoside.
Nucleoside. And when a phosphoric acid group is attached to phosphoric acid is attached to that, we call that as a nucleotide.
Right.
Is it clear? The difference between a DNA molecule and a nucleotide.
Yes, sir.
Suppose I will tell you, give me one molecule of a DNA, right? So what is one molecule of DNA? Now what is DNA? It's a polynucleotide chain, right? Yes or no?
Yes, sir.
Yes,
Yes.
sir.
Okay. So if it is a polynucleotide chain, what should be the length of that chain that we can call that as one molecule? right?
So no definite length.
No definite length. Very good. Then how can we call, like, to what we can call a so listen, the amount of DNA that is responsible for the formation of one chromosome, right? So that is called for a molecule of DNA, and there is no specific length for that. So how many kinds of DNA molecules are there present in case of human beings?
4 types.
No, I told you that each chromosome contains one molecule. So you have 22 autosomes, 22 pair of autosomes. So these are 22 molecules, and then two sex chromosomes, right? X and Y. So altogether there are how many molecules? How many types of molecules? That are 24. Did you get that?
24. Yes, sir.
Did you get that? So for a DNA molecule, there is no specific formula, there is no specific length of DNA. Okay, it depends and it varies from organism to organism. Got it?
Okay.
Okay.
Yes, sir.
So DNA is condensed and wrapped around proteins, forming these chromosomes. And when you will be getting this molecular biology course subject, then you will be able to find out, like, how the chromosome is, what is the structure of the chromosome, and how the DNA is condensed, what is a nucleosome. All these things you will be reading. And this is the DNA that codes for like all these proteins that is synthesized by any organism. Okay, and so we call that this DNA is the hereditary material, because it gives the character of the parents to its offspring. Okay, and they code for these proteins. Now coming to this nucleolus. Okay, so inside the nucleus there is this nucleolus. Sometimes there more than one nuclei can be there, and during the cell division they disappear. Okay, and they are responsible for this formation of this ribosomal proteins. Okay, or these ribosomes, and this is the cytoskeleton, right? See, the cell has a definite shape. Okay, so for a definite shape, just like we human beings, we have a definite shape, and the shape is due to our skeleton, right? If there will be no skeleton, of course the shape what we are nowadays may not be like that. Okay, so depending on the structure of the skeleton, organisms have different shapes. Okay, so in case of this cell, the cytoskeleton is responsible for the shape of the cell. Okay, and this cytoskeleton is made up of several kinds of proteins, microfilaments, and thread-like structures, like made up of actin, and there are microtubules. Those are formed by this tubulin. Okay, tube-like structure. So this is your cytoskeleton, and these are your microtubules. These are your microfilaments. Okay, so just like this, the uh, centrioles, those are present in case of your animal cells, not in plant cells, and those are made up of a bundle of microtubules. Okay, and you can find them during the cell division. Okay, and coming to the centrioles, these are your centrioles, and these are your these microtubules. Okay, this is the kinetochore to which these microtubules are attached during the cell division. Okay, and coming to different, see, today is your introductory class, so I'm not going into the detail of all these things. We'll be just going superficially regarding these organelles, and these things you may not expect in your exam also, theory exams. Nobody will ask you write down the organization of the cell or anything like that. Okay, now this is your mitochondria, and this Altman and Benda, they were responsible for this mitochondrial discovery. Okay, and we call that as the powerhouse of the cell. We can say why we call them as powerhouse of cell.
It
Why?
stores
Store ATP.
Store ATP, sir.
Stores ATP or generate ATP?
store.
Okay, so it generates the cellular energy or ATP. Okay, and there are certain crystals and F1 particles. You will be learning all those things, and of course you know that. See, this is your mitochondria that is surrounded by this double layered membrane, outer membrane. There is inner membrane, and this folding like structure we call them crystal, and this is your matrix. Okay, and it has its own DNA, and this crystal and interior is called matrix. So see, this mitochondria is coming from the egg. Okay, I told you there are certain characters, those are governed by this mitochondrial DNA. Okay, and those characters are inherited from the mother. So if there is any kind of disease that is associated with this mitochondrial DNA, and that is there with the father, it will not be transmitted to the offspring. Okay, but if there are certain disease associated with this mitochondrial DNA, and that is with the mother, then definitely it will be going to the offspring. Okay, so therefore you inherit your mitochondria from your mother, right? So we call them like powerhouse of the cell. These are rod shaped. See, and yes, so these are your ATP molecules, and once this is clipped, and you get lot of energy, and you get this ATP from the metabolism, or this from this glucose metabolism, or this. So this is power plant of the cell, and it burns glucose to form to release this ATP energy. Okay, and it also stores energy as ATP. So next is your endoplasmic reticulum. Okay, this endoplasmic reticulum was first seen in 1945 by this Porter. Okay, and there are two kinds of endoplasmic reticulum, as you know. This rough endoplasmic reticulum that contains your ribosomes associated with that, and these are like hollow membrane tubules, and they connect to this nuclear envelope to cell membrane, and the main function is synthesis of cell product and transport. And this is your rough endoplasmic reticulum. These are your ribosomes associated to the membrane of this endoplasmic reticulum. So we call them like rough endoplasmic reticulum, and they are responsible for the export proteins out of the cell. And see, these are your nuclear core, this is your nuclear envelope, and this is your endoplasmic reticulum. These are your rough endoplasmic reticulum, and these are your smooth endoplasmic reticulum. There is no ribosome associated with that. Okay, so the proteins are made by ribosome on this surface. So they are attached to this endoplasmic reticulum, and they are transported through this uh, endoplasmic reticulum to through these Golgi bodies to different regions. See, though protein is synthesized in your cytoplasm, but the protein they move to different location in the cell, right? Certain proteins they move inside the nucleus. Can anybody tell what are the proteins? Those are moved inside the cell, inside to the nucleus. there are certain proteins, those are synthesized in the cell or cytoplasm, and they are translocated to the nucleus. What are the proteins, or what can be the proteins those need to be transported to the nucleus? Anybody?
Interferon protein.
Yes.
Interferon protein. IF.
Interferon protein.
Yes, sir.
Anything else?
Sir, RNA polymerase.
Very good. DNA polymerase.
Yes, sir. Yes, sir.
All kind of enzymes those are responsible for transcription or for this DNA replication, they need to be transported inside the nucleus. And also, the different kind of transcription factors, those are responsible for these your transcription. These are also synthesized, those synthesized in the cytoplasm, they move inside the nucleus. Okay, the process through which these proteins are transported to different locations in this cell is known as protein translocalization. Okay, or translocation of proteins. We'll discuss in detail regarding how the protein is translocated in the cell. Okay, you see there are certain proteins those are present in this your chloroplast, right? So like this your LSC, this light harvesting molecules. Okay, and complex proteins are there, and different kind of enzyme those are present in this like your it is attached to this end of this rough endoplasmic reticulum. Okay, and it makes the cell product that are used inside the cells. Okay, now these are responsible for making the membrane lipids and regulate this calcium. The storehouse of calcium ion is your endoplasmic reticulum. Okay, and it helps in destroying the toxic substance in liver. Okay, so this is your endomembrane system. This is your nuclear membrane to which this rough endoplasmic reticulum is attached, and after that this smooth endoplasmic reticulum is attached and the distal end. Now, can anybody tell why this rough endoplasmic reticulum is near to the nucleus and the smooth is outside? Because once this your mRNA or this RNA that is coming out of this nucleus, so just after coming out of the nucleus, it can get in touch with these ribosomes. Okay, and this basically this function of these are the smooth endoplasmic reticulum is for this transport. Okay, so these are present at the distal end, whereas these are near to this nucleus. Now coming to this ribosome. Okay, so Faraday discovered these ribosomes. Okay, these are made up of proteins and RNA. Okay, and this is called the protein factory of the cell. You can tell what are the different sites in a ribosome. What are the different sites in a ribosome? Okay, the process by which this protein are synthesized is
The TYC loop.
okay. There is this accepting the side, the E side, right? You may be knowing the existing site. When we'll be discussing this protein synthesis, you'll be knowing that, and these can be attached to this here or maybe like free in the cytoplasm. Okay, so in the next class we'll be dealing a little bit of these organelles, what is there. And before that, I would like to thank you all, and I would like to take the attendance. Okay, I have one attendance sheet, and those are present, please uh, respond to the attendance. Okay, so I don't know. Okay, so uh, please respond if you are there. Okay, of course I can take the screenshot, those are present over there. And please tell your friends that the attendance is a must. If you have more than 95% attendance, as I have told you during the orientation program, you will get 5 marks. If it is not there, then you will not get marks, because these are free marks. So always try to attend the class. Okay, so Kasturi, are you present?
Present, sir.
Oh,
Present, sir.
okay. Mohammad Asadullah.
Present, sir.
Okay. Soumya Chaudhary.
Present, sir.
Sunit Bhushan Patnaik. Sunit Bhushan Patnaik.
Present, sir.
V. Ramaya.
Present, sir.
Pratik.
Present, sir.
Arpita.
Present, sir.
Soumya Rautray.
Present, sir.
Priyanka Priyadarshini.
Present, sir.
Pratyush Kumar Samal.
Present, sir.
Chandan Kumar Behera.
Present, sir. Present, sir.
Shraddhanjali Biswal.
Present, sir.
Sandhya Jena.
Present, sir.
Abhijit Sahu.
Present, sir.
Nigam Shekhar Tripathi.
Present, sir.
Aradhana Satpathy. Akanksha Priyadarshini Nath.
Present, sir.
Monali Manasmita Mohanty.
Present, sir.
Ipsita Behera.
Present, sir.
Garima.
Present, sir.
Jayashree.
Present, sir.
Smruti Lipi.
Present, sir.
Taras.
Present, sir.
Subhrantu Konda.
Present, sir.
Then Subhecha Pradhan.
Present, sir.
Charchika Satpathy.
Present, sir.
Jyoti Prakash Pradhan. Giti Priyadarshini Barik.
Present, sir.
Munmun Acharya.
Present, sir.
Himadri Das Mahapatra.
Present, sir.
Anisha Tripathi.
Present, sir.
Payal.
Present, sir.
ಯಾರೋ ಪ್ರೆಸೆಂಟ್ ಪೂರ್ತಿ
Mrityunjay Padhi. Mrityunjay Padhi. Chandana Dalai. Vishwajit Samantray.
Present, sir.
ಎಸ್ ಸರ್ ಎಸ್ ಸರ್
Chandana Dalai present, sir.
Vishwajit.
ಎಸ್ ಎಸ್ ಸರ್
Murachan.
Present, sir.
Suryakanto.
Present, sir.
Rudra Prasad.
Present, sir.
Naren Basantaray.
Present, sir.
Pragya Paramita Sharan.
Yes, sir.
Ankita Punda, Sagarika Mahanti, Sagarika Mahanti, Payal Rani Biswal,
Present, sir.
Pragya Paramita Ratho,
Present, sir.
Sambit Kumar Sahu,
Present, sir.
Monalisa Naik,
Present, sir.
Supriya Sahu, Supriya Sahu, Satyam Saurabh Samal,
Present, sir.
Satya Sumanta Jena,
Present, sir.
Rosalind Puthal,
Present, sir.
Avinash Kumar Mahanti, Rima,
Present, sir.
Avinash, are you there? Okay, Rima,
Present, sir.
Present, sir.
K. Kanchan, Madhusmita Sethi, Ritanjali Nayak, Shwetarani Das, Sudarshana Ghosh, Priyanka Priyadarsin Nayak, Monalisa Behura, Kirtanan, are you there? Kirtanan, are you there? No. Lule Andrian, and Kalima Godfrey. Okay. Okay, thank you everybody. Okay.
Hello, sir. Supriya Sahu is
Supriya Sahu.
present.
Okay. Supriya Sahu.
Sir,
Anyone else?
Thank you, sir.
Sir, Taras Murmu.
Taras.
Sir, Himadri Das Mahapatra.
Himatri.
Yes, sir.
Himatri. Yes.
Sir, Taras Murmu, sir.
Saurabh?
Taras Murmu.
It's there. It's there.
Payal Biswal.
Which one?
Payal Biswal.
Payal Biswal. It's there.
Sir, Chandan Behra.
Chandan Behera. Chandan Behera is there.
Sir, Nigam Tripathi.
Okay.
Sir, Nigam Tripathi.
Nikam. You are there. Okay. Thank you. Thank you
Thank you,
everybody.
sir.
Thank you, sir.
Welcome.